My name's Steve Strauss. I'm a professor in the Department of Forest Ecosystems and Society at Oregon State and director of the Outreach in Biotechnology program. It's my pleasure to welcome you tonight. This series is sponsored by the OSU Colleges of Agricultural Sciences and Forestry. As the world's population grows and becomes richer, it demands higher quality protein, and fish provide some of the most nutritious and most delicious forms. At the same time, the world's natural fisheries continue to be depleted and degraded by overfishing. A rapidly growing solution to help overcome these converging problems is fish farming, where fish are raised and harvested in confined conditions on land or at sea. Almost universally, the transition from foraging to farming relies heavily on genetics. This occurs both in selection of the species to be used, nearly all of which in all forms of agriculture are exotic rather than natural or local. And it also occurs in the breeding of the selected species for adaptation to the new environment and for high productivity. High yields, of course, improve economics and thus ultimately can reduce the cost of food, but also usually produces undesired impacts such as release of pollutants into the environment. However, yield increases due to breeding also improve environmental performance, such as by producing more product with fewer inputs or less energy, or by using a smaller land or water base. As you'll hear a lot about tonight, the trade-offs are far more complex than the slogans that dominate the headlines, advertising, or activist campaigns. Use of conventional breeding methods to improve yield, today using very intensive DNA analysis to help do the same, raise little concern among the public and is thus essentially unregulated by governments throughout the world. However, when genetic engineering methods are used with the same goal, it generates intense controversy and paralysis. In other words, while science has shown we can do it, society is unsure if we should do it, which is to say that its broad ethical legitimacy is unclear. And the diverse and passionate vested interests and ideologies that are concerned with this decision have created a state of information chaos that is nearly impossible to navigate. I know of no scientist in the world that is better qualified to help us sort through this chaos than tonight's speaker, Dr. Alison Van Eenennaam. Alison obtained her MS and PhD in genetics from the University of California at Davis and her BS in animal science from the University of Melbourne. Alison is an outstanding scientist, having obtained many millions of dollars of highly competitive grant funds for work in animal physiology and genetics, was issued 9 patents, and has published dozens of scientific papers. I am sure that the science she does must be very good because I can't understand even the titles of most of her papers. Despite the esoteric science, Alison is nationally known as the go-to person to help translate animal genetics and biotechnology so that it is understandable and useful. She has produced numerous publications and films to help educate farmers, students, and the public, and has served on several national committees to advise on issues surrounding the regulation and marketing of animal biotechnologies. She was appointed by the Secretary of Agriculture to serve on his Advisory Committee on Biotechnology and 21st Century Agriculture from 2005 to 2008, obtained a National Award for Excellence in Extension in 2010, and obtained 3 Communicator Awards of Distinction for her films explaining animal biotechnology and genetic engineering methods. Since 2002, she has made more than 150 presentations to farmers and the public on the applications of genetics and biotechnology to animal agriculture. In tonight's lecture, titled Ethics of Animal Biotechnology, Should Genetically Engineered Salmon Be Allowed?, to provide context, Dr. Van Eenennaam will first describe traditional animal husbandry and breeding and how it is changing, and explain how genetic engineering is similar and different. She will then focus on genetically engineered salmon as a case study to show how confused the science and laws can become as society struggles to make ethical and legal judgments about it. Please help me in welcoming her to Oregon State.
Thank you. Thanks very much, Steve. I'm surprised everyone didn't run out of the room when they heard that none of my presentation or publications were legible titles, at least. But my objective here this evening is actually to try to make this topic understandable and also to have a little fun. And I have handed out clickers that you all, I hope, have in your hand. And really, the question that's posed here, I'm not going to give you an answer to. I'm going to provide you with information, and then you're going to click and tell me whether you think genetically engineered salmon should be allowed. And so basically, I will go through a little bit these clicker technologies because they're these little boxes. Has everyone got one that wants one? They're anonymous. I don't know which of you has which one. And they kind of look like they might be a little iPhone, but they're not an iPhone. They're not a calculator, and they're not yours. So at the end of the talk, if you wouldn't mind giving them back to the students that are collecting them, and then we'll have them for the next time that I give a talk. But basically, you'll see there's numbers 1 through 9 on there. None of my questions go any higher than 5, so you don't need to worry about anything there. And basically, I will put a question occasionally in the slide deck as we go through, and there'll be an opportunity for you to key in what your response is to the particular question that's being asked. And then in real time, we'll get the answer back. And I know that we've got a range of generations here, which is always fun. And I guess I have kids, and I kind of call them Generation LOL because they're always abbreviating everything, and they love this type of technology. And hopefully some of the older generations that aren't used to this technology will participate so that we can have a look at what the various people in this room think about this particular topic. So with that background then, I'm going to give you a little teaser introduction to the clicker technology just to make sure that we've got everybody using their clickers. And so I'm going to start off with this somewhat shocking slide of genetically modified organisms. And there it is. And I think we all should go home and have a very good look at our dogs tonight and see whether there's any truth to this particular slide. But I'd like to ask you, given this information, which do you think is my dog? And so you have 4 choices there, number 1, 2, 3, or 4. And you'll see that the polling is open here by the little green sign up there. And I can see people are starting to click in there. And so I'm going to give you a little bit of time for this one as people get the hang of what they want to do. This is also kind of how I gauge the mood of the crowd. Because I can kind of get a feel if I'm getting a lot of one particular type of dog, you know, what their attitudes might be about my particular self. And so as everybody that's got a clicker, I should say, if you change your mind after hearing me talk, you might think I'm more like 1 when you hadn't originally thought that, then you can vote again, but it'll only count whatever you vote last as what your final answer to the question is. So with that background then, let's have a look, and I'll have a look. I've got 92 responses keyed in here, and we'll see what the results are. Oh, you're a kind crowd. I thank you, although there are some mean ones in there too. So that little Sheltie down there is my dog. That's Kipper. And I don't know, the other ones were random dogs off the internet. But okay, so I have 67% nice people in the audience. All right. All right, let me ask you a serious question now. Do you oppose the genetic modification of animals? All right, let's see what we got here. That's a pretty typical audience community response. So 68% without any other information saying no. That's a little bit less adverse to the topic than the general Americans. So if you just ask that question to general public America, you'll get about 56% opposed to the genetic modification of animals. Now, What that question means, I have no idea, because I would argue that the dogs on the first slide were genetically modified. And so, you know, maybe what people have in their mind when they get asked that question is something like that's on the screen here. And I love these pictures. I'm a great advocate of using interesting imagery to get your point across, and I think Photoshop is probably about the only place you'd actually genetically engineer these animals. This one is a particular favourite of mine. I don't really know too many species with no alimentary system whatsoever to go through the process of digestion, but this is just basically a head on legs, and very morally repugnant. I mean, there's something very yuck about those pictures, even though it's pretty apparent that it's unlikely that they are actually living in the real world. And so it's really, is it animals that are genetically modified using traditional means that are the problem? And in general, people tend not to be opposed to genetic modifications made by breeding. For example, a lot of them are living with genetically modified dogs that are made by breeding. This is an example of a naturally occurring mutation that's been selected for in certain breeds of cattle. This particular mutation is present in the Belgian blue breed, and it results in this double muscling phenotype, which is for obvious reasons. And basically what's happened there is there's a gene that's mutated, and that gene is called myostatin, and its job is myo, muscle, statin, stop muscles. So if you have two mutated copies, one from mum and one from dad, you get an animal that looks like this. And this is a really popular breed in Europe. Very opposed to genetic engineering, but quite okay with having animals like this that have been bred naturally. And in fact, all of the calves from these particular breed have to be delivered by cesarean section because it's just impossible for the cows to deliver these little muscly calves unassisted. So this tends to be not where we have a lot of issues, but rather it tends to be when you start talking about technologies like genetic engineering and cloning, also called modern biotechnologies. And shown in the slide here is a range of genetically engineered animals and clones that have been produced in various academic labs throughout the world. And they don't look very interesting. They certainly don't look like a canary with feet and a head. They're not particularly dramatic to look at, and yet these technologies tend to engender a lot of concern. And so let's have a little bit of a look at these modern biotechnologies and put them in the context of what we've been doing in animal breeding up until this point. So biotechnology, simply defined, is just technology based on biology, or the application of science and engineering to living organisms. And by that definition, I would argue pretty much all animal breeding and selection is biotechnology. We're using things like performance recording to make selection decisions to improve milk yield in dairy cattle, or marbling in beef cattle, or for selecting for faster growing animals to try to decrease the amount of feed that's needed to produce a pound of beef. Those type of programs, what I would call artificial selection, have been going on. Not a lot of public opposition to that as a technology. Artificial insemination is used extensively in the dairy industry. I think about 80% of all cows in the US are artificially inseminated by semen from very, very elite dairy breeding bulls that are the very best genetic merit that you can have, and that spreads the genetic merit of those animals across the entire national herd. That technology, when it was first introduced, was hugely controversial, and there was concern that there was going to be all sorts of abnormalities associated with those animals. Now it's routine and not something that engenders a lot of public concern. But the genomic technologies that are coming along as a result of DNA sequencing and the genome sequencing that's been going on, and the understanding of DNA, are what's engendered these, what I call the modern biotechnologies. And I'm going to talk a little bit about genomic selection and genetic engineering and cloning to get an understanding of what those technologies are and what they're being used for, and then look at those in the context of the genetically engineered salmon. So I'm going to go through these particular three technologies and ask you to click in, if you will, on your little clicker votes here. and discuss this concept of genomic selection. So this is something that's being used in dairy cattle at the moment, where animals are having DNA taken and they're genotyping the animals, or having a look at what their DNA looks like. And based on pre-existing knowledge of what genetic markers are associated with maybe good milk yield or bad milk yield, or good longevity or poor longevity, they're making genetic selection decisions based on the actual DNA that a cow has inherited, and choosing which animals are going to be the parents of the next generation using this technology of genomic selection. So it's basically, if you will, more sophisticated artificial selection to enable you to better identify with greater accuracy which are the truly genetic superior animals to have progeny in the next generation. So with that little bit of background, I'll ask you what your opinion or overall impression of genomic selection is. All right, I'll shut the polling down there. Okay, 85% favourable, or 84% favourable to that technology. All right. Now we'll move to genetic engineering. And basically, this is defined as using recombinant DNA technology, which is basically a series of procedures that are used in the lab to join together segments of DNA that are going to produce a protein that you particularly want expressed in an animal, and then putting that recombinant DNA into the genome of an animal so that now that animal starts expressing that particular protein. Or maybe you're doing something to actually stop it expressing a protein. For example, the myostatin animal, the Belgian blue double-muscled animal, you could genetically engineer something very analogous by knocking out the protein that is resulting in myostatin. So with that background then, and the discussion of what that technology is, would you say your overall impression of genetic engineering is favourable or unfavourable? Okay, I'm going to shut you down there. Okay, 53% favourable, so we've dropped a little bit in our favourability rating. That one was not as favourable as the previous slide. And then finally, I'm going to talk about cloning. And that's just basically a process that's used to produce a genetically identical copy of an organism. And you're probably all familiar with Dolly the sheep, who's probably the most famous clone that exists in the animal world. And we'll talk a little bit more about Dolly as I get into the talk a little bit more. But I'm just going to, for now, tell you what cloning is. And we'll see what your gut reaction to that is without any information other than that. Okay. Okay, that's interesting. Usually cloning does a little worse than genetic engineering, but so that you were generally more supportive of genomic selection and about even on cloning and genetic engineering. So that attitude ratio is a little bit more favourable than what the general public tends to be with regards to those three technologies. And they tend to be the most negative on cloning. This audience was about equal on that and genetic engineering. But I want to talk a little bit about cloning just to kind of get a feel for where people are on clones, and ask the audience if they've ever eaten a clone. Don't know. Be hard to tell. Might look quite similar to another individual. Okay. 49% of you, I'm sad to say, have probably eaten many, many, many clones today. And that's because clones are used very extensively in plant production. And so if you've had a banana today, you've eaten a clone. Many common fruits and pears and apples and oranges are clones. And they have been produced using asexual reproduction or cuttings from plants. If you've ever done that in your garden, you're actually cloning, maybe not realizing you have been. But that's probably a bit of a trick question, because you didn't think that's what I meant, did you? You thought I was talking about cloned animals. Tricky, tricky. All right. Have you ever eaten milk or meat from a cloned animal? So we'll see if we have much of a change in that particular regard. You have to start clicking faster. I'm not going to give you as long to key in. As soon as I see 80, I'm going to click from now on. All right, let's see what we've got here. Okay, 56% of you are sadly mistaken also, I'm afraid. And let me explain why that might be the case. So nature has been cloning for years without any regulatory oversight. Any identical twin is effectively a clone. What happens there is you've got a developing embryo or egg, and it splits in two, and you get genetically identical individuals or clones. So livestock breeders took a page from nature's book and started using this technique to produce clones in elite livestock, the particularly valuable animals that are used in the breeding sector, and started splitting developing embryos into two or maybe three, putting those embryos back into a surrogate mammal of some sort, in this case a sheep, because we're cloning sheep. They then go to full term, and here you have that sheep that gave birth to genetically identical cloned individuals. And that started in the early 80s. Again, it was used in the elite breeding sector, those very valuable animals. And there's about 2,300 or so that were registered by October 2002. So most likely milk from those animals have been going into the milk supply at some stage. Meat from those animals most likely gone into the meat supply. So you probably have eaten cloned animal products. And that would have been from these livestock that were cloned using this embryo split technique, or maybe even another technique that I will discuss in the next slide. But some of you are probably sitting there and going, that doesn't seem right from a timescale perspective, because I remember Dolly, and I thought Dolly was later than this. And you're quite right, Dolly was later than that. Who remembers when Dolly was born? There was a huge, huge outcry when Dolly was born. And yet, I've just told you that we'd had clones around for at least 15 years before Dolly came on the scene. So why was Dolly such a dramatic event, and why did it cause the concern that it did? And the reason Dolly, the clone, is such an important clone is because she was cloned from what's called a somatic cell that was already differentiated. In her particular case, she was cloned from a mammary cell that came from a sheep. And in fact, that's why she's called Dolly. So the researchers in Edinburgh that cloned Dolly thought that Dolly Parton was a particularly fine example of mammary cells, and that is actually why she's called Dolly. So what happened here was we had the donor sheep here. They took some mammary tissue in this case, and they grew that out in cell culture. Then they took a regular sheep, an egg from a regular sheep, and removed the DNA from the egg. And then they put the diploid cell from cell culture here into that empty egg or enucleated egg and activated that egg so that the diploid genome that belonged to this sheep started developing into an embryo in that egg. That egg again had to go back into a surrogate dam, because that's what mammals have to do to get through pregnancy. And then that lamb gave birth to a genetically identical lamb to the original donor. And what was so unique and startling about this was prior to the advent of Dolly, we didn't understand that you could take a differentiated cell, an adult cell, for example, mammary gland, and actually produce a new individual from it. And that was quite startling to people. And what was really the objective of the researchers, and what was very seldom discussed in the uproar that came after Dolly, was why were they cloning sheep? Do they have a problem over there in Scotland that they needed more sheep? And actually, no, it was nothing to do with that. The objective to cloning differentiated cells was to enable you to do very targeted genetic modifications to these diploid cells, for example, removing a particular gene or adding a particular gene, and then produce an animal from that cell after the genetic modification had been done in cell culture. Now, Dolly herself was not genetically modified, she was just cloned. But that was the reason that people had been so interested in trying to get cloning to be successful, because it enables you to do these targeted genetic manipulations. But when Dolly came on the scene, we had this huge brouhaha that developed because people suddenly realized what that meant was that you could clone differentiated cells from adults, and you might be able to use that on humans. And the whole discussion about animal cloning and the uses of it got kind of tangled up in the ethical debate about human cloning, which is a very, very controversial debate for a variety of reasons. And the ensuing discussion really never failed to elaborate on why Dolly was created and what the application of that particular technology might be for. So that's a little bit about cloning. Let me ask you this. So there's a number of you that have inadvertently eaten animal clones. And now my question is, has anyone here ever inadvertently or knowingly eaten a genetically engineered animal? You're probably soured by this question and you all put yes because you keep getting told otherwise. Let's see. Okay, I might have set that one up for you guys. But 68% incorrect in this case. So there are no genetically engineered animal products that have been approved for human food consumption anywhere in the world at the current time. And that's very different to plants, as you may well know, where we have a lot of genetically engineered corn and soy and the likes. But there have been no animals approved at this stage. And that's actually kind of what tonight's discussion is about a little bit, looking at why that might be and whether that's something that's desirable or not. And so with that background then, I'm going to just briefly go through some of the genetically engineered animals that have been developed and go why they were developed and who developed them, because I think it's kind of an interesting difference from plant biotechnology, where you tend to have large multinational companies. I imagine you can probably guess which one I'm thinking of, that tends to engender quite a lot of public discussion. We don't really have an analogous situation in animal genetic engineering. We tend to have a lot of public sector workers working in this area. And so I'm going to just briefly go through some of the different applications that genetically engineered animals have been developed for. So there's been applications for what I would call environmental purposes. There's genetically engineered animals that have increased disease resistance, genetically engineered animals that have improved product quality for consumption, food consumption, and finally productivity. And therein lies why you're here tonight to talk about the genetically engineered salmon. And so let's have a look at the EnviroPig. That's an environmental application that was developed. And basically, this is a genetically engineered pig that was developed by the University of Guelph that expresses a protein in its saliva that more efficiently digests phytate phosphorus, which is otherwise insoluble in pigs' guts, and goes out the back end. And the back end of pigs is actually quite a polluting problem, and it's this increased phosphorus coming from this undigested phosphorus in pig manure is responsible for a lot of eutrophication and the like. And so it would be very good if we could enable pigs to more efficiently digest their phosphorus and produce less pollution. And this particular individual application reduces the amount of fecal phosphorus coming out the back end of pigs by about 75%. And given the increase in pig numbers that's anticipated as the world population grows, particularly in China, that would be a really desirable application from an environmental perspective. There were mastitis-resistant cows that were developed by the USDA. These are cows that express a protein in their milk that stops mastitis. Mastitis is inflammation of the mammary gland, and it is basically an animal that is resistant to getting mastitis. 2005, that was developed. 2006, we had an application where we have pigs that are able to produce their own omega-3 fatty acids. We know omega-3 fatty acids are good for your brain. That's a lot of people eat fish for that reason. Mammals, and fish for that matter, are unable to make omega-3 fatty acids. That's the purview of certain algae and organisms like that. And so this particular application used cloning to make a genetically engineered cell that had the the protein that they want expressing, they use that cell to then clone these pigs, and that's why you see that they're a generation of cloned transgenic pigs that are able to make omega-3 fatty acids. Developed in 2006 by the University of Missouri and Massachusetts General Hospital as a healthy pig product, if you will. And finally, in 2011, just this year, there was a very interesting development, again out of Roslyn, home of Dolly, and that is the development of genetically engineered chickens that are unable to transmit bird flu from bird to bird. Well, you might say, well, I don't care about bird flu. Well, let's have a little bit of a think about bird flu. The last time that bird flu became human virulent, so it crossed the species barrier from chickens, avian influenza, to humans, and then it mutated to become human-to-human transmissible, such that I could give it to you rather than a chicken. We had about 20 million people that died from that flu epidemic in 1918. And that was in the days when we had very little air travel and very little international commerce of the likes of which we have today. If we were to have a bird flu that crossed the species barrier and was actually became human-to-human transmissible, it would be a very big public health problem. And I think the 20 million would be chicken feed, so to speak, relative to what would happen if that was to occur today. So let me ask you this question, and that is, if there was a way to engineer resistance to influenza in poultry, should we use these genetically engineered disease-resistant chickens to produce our poultry meat and eggs? And so we have the technology developing to have these genetically engineered chickens that are avian flu resistant. Is that an application that you think you would be supportive of in using genetically engineered animals? Okay, so that's a bit of a shift from the previous feeling on genetically engineered animals. And so if there's a benefit associated with the use of them, there's some flexibility there in terms of people's opinions. Some people still think no, they're not, 25% are still not interested in using genetic engineering for that particular purpose. I'm going to give you a fun case study now, or I think it's a fun case study, and it's got to do with mastitis. And I like this because I actually had mastitis when I was nursing my kids, and so it's a very personal example for me. And I think it's always fun to look at a nursing mammal, and that really does engender everything good about motherhood. But mastitis, not so much. And for those of you that haven't had the joy, it's basically when bacteria get into your mammary gland and cause inflammation and infection, and you have to do something about it. So I'm going to present to you 5 different options for dealing with mastitis in dairy cattle in this case. And it's one of the biggest disease problems that there is in dairy cattle. And so at the moment, the traditional way, the conventional way of dealing with it, both at a human medicine level and also on a conventional dairy farm, is to treat either the patient or the quarter, in the case of dairy cows, with antibiotic to kill the bacteria that have invaded the gland. So that's my first option for you. My second option is to use a natural or alternative therapy to try to deal with mastitis. And so I looked through this book called Natural Cattle Care to see what the suggestion is if you're not using antibiotics to deal with mastitis. And this book suggested that you should give an extra tablespoon of dolomite, which is, I believe, a type of soil, night and morning until the infection clears. Or you could squirt 10 mils of hydrogen peroxide straight into the affected quarter. So that's alternative 2. The third alternative gets back to one of the modern biotechnologies I alluded to earlier on in this talk, and that is the use of genomic selection. And you can actually, by tracking the health of cows in the national herd, we can start to identify which parts of the genome are associated with mastitis-resistant and basically predict the genetic merit of these highly used sires to predict the sires that are least likely to transmit mastitis susceptibility or most likely to transmit disease resistance to mastitis. So we could use that biotechnology to select superior sires. I'm going to put up there the genetically engineered cow that's able to basically be disease resistant as the result of a production of a protein in its mammary gland that's genetically engineered. And then finally, I'm going to give you the option to clone a bull of all of whose daughters have proven to be resistant to mastitis. And so clearly he's got good mastitis resistant genes that he's going to pass on to his offspring. So with that background then, and this is always a fun result to see because I have no idea what I'll get here, but you basically have the 5 choices here of how you want to deal with the problem of mastitis on a in the dairy industry, I'll say more generally. And I'll give you a bit of time to think. Oops, well, maybe I won't. Sorry about that. Okay, interesting split, almost straight across the board there. And interestingly, well, I don't know. You have to look at that and think about what you think about that particular response. I almost always get someone that responds in every category when I ask this question in public audiences. Not usually quite as even a split across there as is seen in that particular one. Okay, and so we are actually getting what, 19%, so almost 40% that were voting for genetic engineering and cloning there in that particular application. So giving you a bit of background there, let me just ask you a very simple question. Would you eat Frankenfish? Like I say, I think some of the images that you can find on the internet, I just actually Googled AquAdvantage salmon in Google Images and got this picture back. And it's a very creative picture, and I think kind of an interesting. I guess if I see the term Frankenfish, if you really want to annoy me, use that term because it's so pejorative. You know, it's how can you have a rational discussion after calling something Frankenfish? But anyway, all right, everyone keyed in that wants to? Okay, 65% opposed to Frankenfish. That's a little lower than the general public generally is, especially if that's the only information they ever get. So with that background that I've given you to date, then let's have a little bit of a discussion of genetically engineered salmon, and we'll kind of move on to fish here. So let's talk a little bit about aquaculture first, and salmon aquaculture particularly, so that we can start to have a look at where the genetically engineered salmon is envisioned or would likely fit in this. And I think one of the things that was alluded to in the introductory comments is the increasing demand for animal protein in the world. And in 2006, about half of the world fish food for humans was coming from aquaculture, and we need to increase that amount by about another 30 million metric tons by 2030 to keep up with projected population growth if our choice is to continue eating fish. And that is something that has been seen to be what people are interested in eating as if their income starts to increase, or at least animal protein does tend to be one of the first things people do as they move out of poverty. And in the developing nations, we certainly are seeing huge increases in India and China with regards to their demand for animal proteins. And so aquaculture continues to grow more rapidly than any of the other animal food producing sectors, so beef and sheep, even pigs and chickens. And worldwide, the sector has grown about 8.8% per year since 1970. Compared to only about 1.2% for capture fisheries or wild caught fisheries. And so the difference between what we're currently producing and eating versus what's projected to be produced and eaten has to pretty much come from aquaculture if we're going to keep up with that demand, because the wild caught fisheries are not increasing the amount that they're producing, and in fact there's a number of them that are in very severe decline as a result of overfishing, depending upon how their population's being managed and what sort of fishing practices are being implemented by those fishermen in the wild caught fisheries. And this is not just America, but throughout the world, of course. There's fishermen in a number of different oceans. So there's going to be an increased need for aquacultural fish production if we're going to produce enough fish to feed the projected population increases. Let's have a look at salmon, and I know that's a topic that's probably dear to many people's hearts, or you wouldn't be sitting out here at 7 o'clock on a Tuesday night. So let's have a look. In 1996 was the first year when aquacultural production of salmon exceeded the wild catch of salmon. Since that time, it's been more than the wild catch of world salmon. And in America, and very few people know this, almost all of the Atlantic salmon that is consumed in America is farm-raised Atlantic salmon and imported from either Canada, Chile, Norway, or Scotland. They're the big four that are producing aquaculturally raised Atlantic salmon. And if you look during the years of 2000 and 2004, which was the last time I could get good reliable figures, Americans were basically consuming 0.28 million metric tons of salmon, and we're actually pretty lightweight in terms of our fish consumption compared to some other countries where it's a lot more an important part of their diet. But of that, in general, about a third of that was Pacific salmon and 2/3 was Atlantic. 1/3 was wild, that was pretty much the wild-caught Pacific salmon, and 2/3 was farmed, that was pretty much the Atlantic salmon. 1/3 was domestic production, wild-caught Pacific salmon, and 2/3 was imported, again, the farm-raised Atlantic salmon. And one thing I do just want to bring up, because sometimes there's some confusion here, is Atlantic salmon is a different species to the Pacific salmon, and they cannot interbreed with each other. And I realized when I got off the plane that obviously salmon was very important to Oregonians, because there was a pillar at the airport just as I got off the plane, and this was the pillar that I saw there, and it had the story of salmon kind of engraved into it, and I thought, wow, there's a good educational tool there sitting at the airport. So here's the salmon family of Salmonidae. Pacific salmon are the genus Oncorhynchus, and there it lists the vast variety of all the different species of Pacific salmon that you've heard of, no doubt. Things like sockeye and coho and silver, and the vast variety of salmon that exist in the Pacific. And they're quite different from the Atlantic salmon, right, which is a different genus altogether. It's not even Oncorhynchus, it's Salmo, and they live in the Atlantic. They're Atlantic salmon. And actually, there's no really wild, American-produced Atlantic salmon really on the food market, because these are basically an endangered population. Mostly, this is a picture of Maine here, and this is the critical habitat that they're trying to restore to enable those wild populations to get back from basically being an endangered species. And so there's quite a difference between the two, and I just want to make that point. And I also want to make the point that if you go to the supermarket, and I'm going to show you a picture from Davis here, because that's where I live, and this was some market research, for want of a better term, where I just went to the store where I go shopping and had a look what there was available in terms of salmon. And basically there was the Atlantic fillets over here that are farm-raised, imported Atlantic salmon fillets. And they were being sold for about $7 a pound in this particular Safeway, that if you had your club card with you. Okay, so that's what it is, and of course it's labelled because it has to say that it's imported and farm raised, and so you know what you're buying, and that was one option. And the other option that was available was wild-caught Pacific salmon, in this case king salmon fillets, which was about $18 a pound. And that was what it was in the marketplace when I went and looked at my particular store. So 2/3 of the salmon eaten in America are these, 1/3 of the salmon eaten in America are these. Let's talk a little bit about aquacultural production practices. Because there's basically, I would argue, 3 types of salmon aquaculture. The most common approach to raising salmon, particularly Atlantic salmon, is this net pen aquaculture, such as shown here, where you've got the animals in pens in the ocean and they're getting their feed in these net pens, and there's some environmental concerns associated with that particular type of aquaculture pertaining to environmental degradation under the net pens and the potential transmission of diseases both from wild salmon populations to the caged populations and vice versa. So they can interact with each other. And there's also some concerns about disease transmission and sea lice and that type of thing. And so there's a lot of opinions about whether or not aquacultured salmon is a good thing. And so this is net pen aquaculture. A second type of aquaculture is what I call sea ranching, where the salmon eggs are raised in a hatchery to produce a lot of young, and then those young are released into the ocean, where they forage in the ocean, and then they're wild caught, and they can be sold as wild caught salmon. And in 2008, it was estimated that about 41% of the Alaskan wild salmon catch were probably hatchery-raised fish in their juvenile stages, but then they go out and grow to a catchable size out in the ocean. So that's the second type of aquaculture. And then the third type is a relatively new concept where to kind of avoid a lot of the problems associated with aquaculture in sea pens, you bring the fish on land, but you still have to put them in water, so you put them in inland tanks. And so this basically involves keeping the fish away from the native populations, so you get rid of that disease transmission either way, and parasites and that type of thing. And they live their entire life in these contained tanks. And you can actually have fully recirculating systems so that the water is going around and around. It's not a flow-through system. So you can have relatively water-efficient aquacultural tank systems. And they can either be fresh or salt water. And in the case of Atlantic salmon, you can grow their entire life without salt water. That's not the typical life course of an Atlantic salmon, which typically would smolt and go to the ocean. But they can live in these tanks. And you can basically take them through the tanks, harvest them from those tanks, and then they go to market from there without ever having any interaction with the ocean. And this is called land-based aquacultural systems. So let's have a little bit of a talk about the salmon. So we've heard about the AquAdvantage salmon, and it's a fast-growing salmon. And I just draw attention to this paper because this is the paper that first described the AquAdvantage salmon. And you'll notice that the publication date on this paper is 1992. And in fact, the original founder fish, that is the first genetically engineered fish that carried this genetically engineered construct, was created 21 years ago. And this particular fast-growing fish has been trying to get through to market for over 20 years at this stage. And so let's have a little bit of a look at what I'm talking about when I'm talking about this genetically engineered AquAdvantage salmon. And I know you're not all familiar with genetic engineering, and so I'll try to explain this in a way that's comprehensible. Basically, recombinant DNA techniques in the lab were used to isolate DNA from the ocean pout fish, and particularly the little DNA segment that was taken from that fish was an on switch that turns genes on and off. It's called a promoter, and this particular promoter in the ocean pout drives the antifreeze protein, which turns on in the winter. So this was an on switch that turns on in the winter. That was hooked up to the growth hormone from Chinook salmon, okay? And so now you have a growth hormone that's going to be under the control of an on switch that turns on in the winter. Okay, with me so far? So you hook up those two together, then that was injected into the eggs of Atlantic salmon to produce Atlantic salmon that are carrying that DNA construct, this growth hormone under the control of the promoter from the ocean pout, to produce the AquAdvantage salmon. That is a salmon that grows not only during the summer and spring when typical wild salmon would grow, but also grows during the winter as a result of this additional transgene. And typically in the native environment, you wouldn't want to be growing in the winter because there's not a lot of food around as a result of cold water temperatures. But however, if you're growing animals in aquaculture, ideally you'd like animals to grow the whole year round so that you can market them as soon as possible, because then you're going to have less feed going into them to produce a market weight fish. And so these fish are able to reach adult size in about 16 to 18 months. Typically, a salmon is going to market at about 2 kilograms. And so they're able to get there in about 16 to 18 months, instead of the typical 30 months that it would take for a non-transgenic, a standard salmon, to reach that market weight in an aquacultural production system. And that's very appealing to fish farmers, because feed is about 70% of the cost of producing a pound of saleable fish. And the quicker you can get an animal to market, the more cost effective it is to raise that animal. These fish don't look different to a regular Atlantic salmon. This is a set of siblings. One is expressing the transgene. I'll let you guess which one that is. One's not. But they're not looking different. They just get to market size in a quicker time. And they don't get twice as big or 5 times as big as a regular salmon. They just get to mature size more quickly, and then they go to market more quickly. So the company was interested in marketing this fish when they first developed it. And so in 1993, so just to put time frame back, the fish was created in 89, and then that publication in Nature came out in 92. And the year after that, the company went to the FDA and said, we'd like to bring a genetically engineered fish to market. How do we do that? And the FDA went, I don't know, no one's ever done that before. We didn't have the precedent of the plants to fall back on. And so the FDA had to make a determination, well, how are we going to allow these genetically engineered fish to reach market? How are we going to regulate these fish? And so 15 years later, the FDA came back with their determination of how they were going to go about regulating genetically engineered animals. And basically the decision they made after extensive consultation with scientists, public interest groups, other government agencies, was to regulate these animals as new animal drugs. And that sounds very strange. And if you're interested in understanding a little bit more about that decision and why that was made, I have here a CAST report on the science and regulation of food from genetically engineered animals, which goes into detail as to why that particular approach was used and some of the pros and cons associated with using that particular approach. The pro, or the advantage of using that particular approach, is it requires a mandatory evaluation by the FDA before that product, in this case the drug of a genetically engineered fish, can go to market. And that means that it has to be looked at from a food safety perspective. It has to be looked at from an animal safety perspective. Is it safe for the fish? It has to be looked at from an environmental perspective. And it has to be looked at to make sure that the fish or the drug does what the sponsor says it does. And in this case, the sponsor said it grew more quickly. And so that was basically what the FDA laid out in terms of how you would bring a product to market. You'd have to prove to their satisfaction that it was safe for the environment, it worked, and it didn't harm the animal. And so with that background then, in 1995, AquaBounty first submitted an investigational new animal drug application in which they said, we plan to bring this product to market. And 15 years later, the FDA finished its review of the data it had collected on food safety and various, whether the food had the same components, whether there was anything abnormal about the fish. And they presented this data at this Public Veterinary Medicine Advisory Committee meeting that was held in September 2010 to consider the safety and efficacy of the AquAdvantage salmon. And basically, what the product that the company was looking to get approval for is outlined very simply on this slide. And basically, I want to draw your attention to a couple of things about what the company was proposing or is proposing. And the product identity says that they wanted to get approval for a triploid hemizygous all-female Atlantic salmon with a single copy of the transgene. Now, I know that's a mouthful, but what that more or less means is that the fish that they're seeking approval for are triploid, And triploidy in fish induces, and any species, induces sterility as a result of, it basically messes up the meiotic disjunction in the development of oocytes and gametes. And so they were looking to get approval for a sterile, all-female Atlantic salmon product that would be bearing a single copy of the transgene. They said that it grew significantly faster than its non-transgenic comparators. And most importantly here, the proposal was to produce these fish in FDA-approved, physically contained freshwater culture facilities, i.e. land-based aquaculture, the tank system I described, not in ocean net pens, where it's known that there are escapes into the wild population. And particularly for this application, it was only to grow these fish in Panama. And this is the facility that the company was looking to get approval for. And basically, this is in the highlands of Panama. And there was a number of different containment approaches, physical containment, things like stopping birds having access to the fish, nets and various things to stop any of the life stages from escaping. And the location in Panama was such that if a fish did get out of that facility, there were a number of thermal lethal temperatures in the waterways leading to the oceans, and including 2 electric or hydroelectric dams that really would preclude any fish from making it to the ocean if, in the unlikely event that it got through the multiple redundant containment measures that were in place on these land-based aquacultural facilities. And the rationale for this is really, and I'm going to list here what the potential advantages of this land-based inland tank AquAdvantage aquaculture concept is, what the company is trying to get done. And basically, as a result of the fact that those fish grow that much more quickly, you're able to get the fish to market in a shorter production time. I've already mentioned that, 16 to 18 months versus 30 months. And that has a number of benefits, not the least of which it's less expensive to get fish to market, which enables you to then have some money to invest to build these on-land recirculating tanks. So they're not inexpensive to do this. And so the concept behind having the fast-growing fish was to enable it to be financially viable for you to produce land-based aquacultured salmon in a less environmentally degrading way than ocean pen aquacultured salmon. And so the fact that they have the improved food conversion ratio, that is, they produce more product per pound of feed, is really what drives the economics of this to enable this type of what some might argue is more sustainable and ecologically friendly salmon aquaculture. And although the application that was put before the FDA was for this facility in Panama, if they did want to do anywhere else, they'd have to submit another application to the FDA. But conceptually, and this is not what they were proposing, but I just want to bring this concept to you because it'll play into my last clicker question for you, is conceptually, if you've got triploid all-female fish that are biologically contained, there's no reason why you couldn't have a freshwater tank system set up in the heart of downtown Los Angeles, and therefore be able to actually produce salmon aquaculture in the garage of the Sears building or something along those lines. And it potentially opens up the possibility of American-raised Atlantic farmed salmon that could be used to provide the local market with the salmon that it needs, because it removes the need to be located close to the ocean from aquacultural production of salmon. So you know what my last set of clicker questions is going to be, don't you? So I'm going to give you a case study of salmon and ask you which of the 4 following types of salmon you would buy. You've got your wild-caught, $18 Pacific salmon, at least in Davis market there, that are coming from the wild-caught fisheries in Alaska and off the coast there. You've got the current situation if you're buying Atlantic salmon in the US, and that's an imported product that's been raised in these net pens off Norway, Scotland, Canada, and Chile. I'm going to put that at the $6.99 that they were at my Safeway. And then I'm going to give you two options that involve the genetically engineered salmon. And I'm going to say, as a result of the investment in the recirculating system, that the price doesn't differ from traditional farm-raised salmon. But the advantage, or the difference here, is you've raised the salmon in a more sustainable land-based system without a lot of the ecological concerns associated with the net pen salmon. And then finally, this is a very hypothetical one. I've got exactly the same scenario, but you raised it here in a garage right in Corvallis. And so you've actually got locally grown farm-raised Atlantic salmon available to you. So with that background and that introduction, which ones would you select? And I'll give you a bit of time to think about that. Okay, I've got my magic 80. Last few stragglers. Everyone voted that wants to vote? All right, what do you guys reckon the answer's going to be? Let's have a look. Interesting. So kind of evenly matched on the wild or genetically engineered, number 4. I guess if we looked at the GE Panama and added those two together, we got 49% that are voting for genetically engineered aquacultured salmon, 39 going for the wild, and 13 staying with the current status quo. So there's a number of, I forget what the actual number is, that said that they wouldn't eat Frankenfish, but I think there might have been some that have changed their vote there and have looked at the big picture and said that in their particular buying decision, that they would support an approach like that. Well, I'll just finish this story because the saga's not over. So September 2010, there was that public meeting where the FDA was to consider the application of the salmon coming to market. And for these 49%, I'm not sure you're going to have that option, and I'll tell you why. So this is my little timeline here. We've already discussed that it's been 15 years in regulatory evaluation. About a year after the public meeting where the data was presented, the House of Representatives passed a vote to prohibit the FDA from approving any application of genetically engineered salmon. And so we actually had a representative from Alaska and California, and they argued that these fish would create competition for the state's fishing industry. And in the statement, this representative said he had concerns about the fish, which he calls Frankenfish. Kind of tells me where he feels on that scale. And in this particular letter, he suggests that Frankenfish is uncertain and unnecessary. And basically the amendment, so defunding the FDA from doing the drug approval process on the fish, would stop them from being approved because they seriously threaten wild salmon populations because they grow twice as fast and they require much more food. Okay. Don't think there's much science to support that they need more food to produce a pound of feed, but subsequent to that, in July of 2011, there's been a number of senators from various states, I think including one from this state, who have written a letter to the commissioner urging her to stop evaluating the salmon, not cease and desist, if you will, from approving this new animal drug application. At the current time, the senators, so that's the upper house, have not voted on this budget amendment, and so it hasn't become law yet, because you need both houses to pass that for it to come into play. And so we're kind of at an impasse because it's not clear what's going to happen, to be honest with you, whether it's going to be approved or whether it's not. It's been over a year since the public meeting, and I don't think anyone knows what the current status of the approval or not approval is. And I think this particular political action, after 15 or so years in the regulatory process, to me is perhaps almost the most concerning part of the whole story, because it's not just this particular fish that's at stake in this case. So it's really the principle behind the amendment, and that is, I don't like this drug that you guys are regulating. I'm not going to give you any more money to approve it. If we start applying that criteria to other new animal drugs or human drugs, I wonder if we would have, for example, maybe the morning after pill approved, where you again have political sides that maybe are not supportive of that use. Maybe the contraceptive pill might be next. There's various ethical issues that certainly come into FDA decisions regarding the use of drugs. However, historically, the US has had a science-based regulatory system, and it really has served the country well. I know thalidomide is something that's near and dear to my heart, because in Australia that was approved for morning sickness, and my brother's 4 years older than I, and that was the age group that got hit by thalidomide, which causes severe birth defects. The FDA did not allow that product to go to market. They have a very rigorous science-based review process in place. And if we're going to say now that you can go through a regulatory review and then at the last minute have political interference that stops a product coming to market, would we ever have any products coming to market? And maybe you don't like the fish, but what about the genetically engineered disease-resistant animals or other applications that are very socially redeeming, if you want, for, that would have broad public support? What company would want to go through the type of regulatory problems that is being experienced by this particular application? And I'll just close with this last slide because I think it's interesting. I was at a conference on international work on transgenic animals, and Europe got up and America got up, and we had our discussions about all of the opposition of various groups to this technology, and then this Chinese researcher got up and just listed off about 30 different applications that Chinese researchers are working on, and had their trajectory of when these different applications were coming to market. And what was kind of distressing to me a little bit was every single application he had up there was technologies and applications that had been developed in the US and Europe and Canada, and the exact same application had been made in Chinese animals and are now proceeding forward to market. So we had basically mastitis-resistant pigs that were expressing a protein in their milk to resist mastitis. We had transgenic pigs expressing phytase in their saliva. Well, that's the enviro pig in Chinese. We've got pigs that are resistant to foot and mouth disease, which is a big issue. And then here we have omega-3 transgenic cattle that are expressing the omega-3 producing proteins and would be able to be land-based sources of omega-3 fatty acids. And so I think that's kind of interesting, because I think that country is, with its huge population and huge demand for animal protein, is going to go forward and use this technology at least to feed its own people. And then the exports will be a different story. But with that, I'd finish, and I'm very happy to take questions.